Ladies and gentlemen, hello and welcome to Experiment 1 of Chem 6CL. The purpose of this lab is to observe how purple cabbage can act as a pH indicator, and our final objective is to understand on a molecular level what we are observing. To start, let's take a look at the required materials. Really briefly, we have materials, so you're going to need a purple cabbage head, of course, and then one knife, a cutting board, and then one pot for the boiling method and one blender for the blending method, one sieve to remove the solids, and 3 beakers and cups. It'd be see-through or glass. And then the acids and bases that will be used to change the pH here. And you can find the table in the procedure that I'm going to go over next. It first starts with taking your cabbage and cutting off the stem with a knife on a cutting board. This will make it easier in step 2 when you start to peel the cabbage leaves off until you have about 1 bowl's worth. The step 3 and step 4, these are 2 methods and you can choose either way depending on the resources available. So in step 3, you're going to blend it and you're adding your bowl's worth of cabbage leaves to the blender vessel, and then add 2 cups of water and blend until you see the dark purple color. And then once you see that dark purple color, you can turn off your blender and use a sieve to separate the solid from the liquid. But what you want to keep is the liquid because that contains the liberated molecule from the purple cabbage. And you're going to use that as your pH indicator. So in the boiling method, you're just going to add the 2 cups of water into a pot and place it on the stove, turn on the gas, and then turn on the heat to about medium-high and bring the water to a boil. Then once it's boiling, you can add your bowl's worth of cabbage into the pot and submerge it completely in the water. Then simmer for 5 minutes or until the water becomes dark purple, and then turn off your stove, remove from heat, and cool until it's lukewarm. This is so that you can filter out the cabbage pieces and again keep the purple extract. Then in step 5, there's plenty of acidic and basic substances in the house that you can use. So once you've identified which of the substances you're going to use, you can move on to step 6, which is obtaining 3 beakers and label them acidic, neutral, and basic. Then you're going to add approximately 1/4 cup worth of your purple cabbage extract to each cup. And then you will add your acid to the beaker labeled acidic and your base to the beaker labeled basic. Then, because this is a pH indicator, you're going to see color changes, so you should observe and record the results for those color changes. Okay, so now that we have seen how the process is done, let's quickly go over some discussion questions about what we observed and why. So the first question is, why do certain compounds have color? And this was discussed in the methyl orange lab, where chromophores are the parts of compounds that can absorb UV or visible light. So if a compound contains a chromophore, it has the potential to absorb and emit certain wavelengths of light, and it does so by absorbing the energy of a particular wavelength of light. And this particular wavelength of light has a certain frequency or certain energy that's associated with it that can take electrons and promote them from the HOMO to the LUMO, the HOMO being the highest occupied molecular orbital and the LUMO being the lowest unoccupied molecular orbital. So when the compound absorbs this visible light, so say this green light is coming in and it's the right wavelength to promote these electrons to the antibonding orbital, then it emits a color that is complementary to the color absorbed. So if it absorbs green, it emits red. And then the compounds can emit different colors based on the amount of conjugation present in the molecule. So having conjugation decreases the amount of energy needed to promote an electron from the HOMO to the LUMO by raising the energy of the HOMO and lowering the energy of the LUMO, which is what's seen here. So the energy needed here is much greater than the energy needed here. So in a molecule with more conjugation, it requires less energy. And if we follow on the equation of energy is equal to Planck's constant multiplied by the speed of light divided by wavelength, then we can see that as energy increases, wavelength decreases, and as energy increases, or as energy decreases, wavelength increases. And so we can follow that up for question 3, but question 2 is asking, what is the name, class, structure of the molecule we are extracting and using? So the name is, it is an anthocyanin, and it is part of the flavonoid class of compounds that are typically found in plants that are blue or purple. So that's why we use purple cabbage. The structure looks similar to the one presented here, but there are different groups that can be substituted. But this is the easiest molecule for the explanation. So question 3 is bringing it all together to explain how it works. So as a reminder, this is what our final result looked like, where the acidic was pink, the neutral was purple, and the basic was blue for the most part. So we can use this image to kind of explain that depending on the pH of the solution changing listed here, the structure will change, and the structure changes the conjugation, which changes the wavelength absorbed and the wavelength emitted. So we see here first on the right, basic conditions. So basic conditions, you can see that this hydroxyl group was deprotonated, and it has a carbonyl and then 3 other OH groups. So this oxygen can delocalize its electrons into the ring and have extended conjugation. And so this molecule is we would say the most conjugated, or has the most conjugation. And because of that, it absorbs the largest wavelength of light, which is around this area here, the orange, and therefore emits blue. In contrast, violet, at neutral pHs, the compound is violet, and that's because it's very similar to the basic structure. However, this hydroxyl group is protonated, and that slightly decreases the amount of conjugation that the molecule has. Which is why, so if it emits violet, then that means it absorbs wavelength, the wavelength of yellow, which is slightly higher in energy than orange. And finally, the last compound, we can see that the carbonyl group has turned into another hydroxyl group, which overall decreases the amount of conjugation even more compared to the neutral. And so it emits red light. And if it emits red light, that means it absorbs green, which out of between orange, yellow, and green, green is the highest energy and the shortest wavelength. So the conjugation can explain these different color changes. And even further, we can have, upon very small changes in pH, the color can change. The color can change not as drastically, so you can even measure the difference between pH of 2 versus pH of 4, rather than in cases like methyl orange, where they were kind of polar. We have very defined separation depending on the pH here. So we can get a whole rainbow of colors, and yeah, so that's all I have. Thank you so much for listening, and I hope you enjoyed the video.